Assalamualaikum, we are from Group 5. We'll be presenting about protein expression.
Okay, so what is protein expression? So protein expression refers to the way in which proteins are synthesized, modified and regulated in living organisms. Proteins are synthesized and regulated depending upon the functional need in the cells. So the blueprints for proteins are stored in DNA and decoded by highly regulated transcriptional process to produce messenger RNA or mRNA. So the message coded by an mRNA is then translated into a protein. So transcription is the transfer of information from DNA to mRNA, and translation is the synthesis of protein based on a sequence specified by mRNA. So that's about the principle of protein expression. So there are two principles of protein expression. First, in eukaryotes. In eukaryotes, the process are spatially separated and occurs sequentially with transcription happening in the nucleus and translation occurring in the cytoplasm. After translation, polypeptides are modified in various ways to complete their structure, designate their location, or regulate their activity within the cell. So next, in prokaryotes. So in prokaryotes, the process of transcription and translation occurs simultaneously. The translation of mRNA starts even before a matured mRNA transcript is fully synthesized. This simultaneous transcription and translation of a gene is termed coupled transcription and translation. So that is the principle for protein expression in prokaryotes and eukaryotes.
Elements in protein expression vector. First element is the promoter, which is the most essential element in the protein expression. The function of the promoter to ensure a reliable transcription of the gene interest. Furthermore, the strong promoters are also important for an efficient mRNA synthesis with RNA polymerase. Second is terminator, which acts as gene transcription termination. Additionally, it also functions as mRNA stabilization. Next is ribosomal binding site, or known as RBS. It follows the promoter that is responsible for the efficient translation of the initial site. The translation initial site in case of prokaryotes is known as the Shine-Dalgarno sequence. Meanwhile, the translation in eukaryotes is initiated at a particular sequence called Kozak sequence. Then, another element is selectable genetic marker, which allow me identify bacteria that have successfully transformed or taken up the recombinant plasmid. Antibiotic-resistant genes are also commonly used as selectable markers. Protein tags are also one of the elements in protein expression. It acts as convenient tool which improve the solubility of recombinant proteins and streamlining protein purification. Furthermore, it utilized to easier track proteins during protein expression and purification. Last but not least is multiple cloning site, MCS. MCS is also called polylinkers, which allow for the insertion of foreign DNA without disrupting the rest of the plasmid. In addition, the regions are 50 to 100 base pairs in length.
Choosing the most appropriate expression system is one of the most important factors to consider. So this is because a poor choice can essentially lead to poor expression yields or to inactive protein expression. So the first factor need to be considered is time spent for expressing the protein. As we know that different expression system takes different time to express the protein, and usually our aim is to express a protein in a short time. As shown in the table, bacteria act as a rapid systems of expressing protein due to the short doubling time, which is 20 minutes. The second factor need to be considered is easy handling of the protein expression system. So bacteria is one of the easy expression system because it's easy to culture, media required is not expensive, and also the method adapted to scale up bioproduction are straightforward. So compared to mammalian expression system, it requires continuous carbon dioxide supply and also require expensive transfection reagents. The third one is post-translational modifications, or PTM. So usually after translation, polypeptides are modified in various ways to complete their structure, designate their location, or regulate their activity within the cell. So PTM are various additions or alterations to the chemical structure and are critical features of the overall cell biology. So for example, bacteria lack many of the PTM present in eukaryotic cells. Therefore, when we want to produce eukaryotic proteins that require PTM, a mammalian expression system is the optimal choice. The last factor need to be considered for the selection of protein expression system is the structural complexity of a protein. So the bacterial expression system is the clear choice for production of prokaryotic protein and also ideal for eukaryotic protein with a simple tertiary structure that contains a single structural domain. However, eukaryotic proteins with complex tertiary and or quaternary structures are usually best suited for insect or mammalian expression system. This is due to it is equipped with the machinery for proper protein folding and PTM.
The first types of protein expression system is bacterial expression systems. It is a rapid and simple system of expressing recombinant proteins due to the short doubling times. The most widely used host system is E. coli, since there is ample knowledge of its genetics, genome sequence, and psychology. The genetic manipulations is easy and is also grows in high densities and is suitable for large-scale fermentations. However, the cell wall of E. coli contains toxic pyrogens, and the expressed proteins may have to be extensively tested before use. For yeast expression systems, it is a highly developed genetic system, as Cerevisiae is an attractive organism for the expression and production of recombinant proteins. Yeast able to carry specifically designed plasmids. This ability is valuable in a recombinant protein expression systems. The plasmid use consists of restriction sites that can be used to insert gene sequence of interest, and the transformations of yeast with the plasmid produce the desired protein and can be appropriately scaled up. For mammalian cell expression systems, this system is an ideal system to express the mammalian protein that require multiple post-translational modification for the production of functional proteins. It can produce transient or stable expressions of proteins in cell lines. Cell lines such as HEK2 and CHO have been developed as efficient, transient, and stable expression systems. HEK2 cells transiently transfected using lysosomes, calcium phosphate or PEG as transfection reagents. CHO commonly used to stably express large quantities of recombinant proteins, and this process involves transfections of DHFR deficient CHO cells with the gene of interest and a DHFR selection cassette. Insect cell lines adapted to serum-free suspension culture. The SF9 insect cell line is a clonal isolate derived from the parental Podoptera frugiperda cell lines, while SF21 insect cell line is derived from the ovarian cells of Podoptera frugiperda. For cell-free expression systems, the proteins assembled in vitro in a single reaction using cell-free reaction mixture. The mixture includes transcription and translational machinery. This system is suitable for fast assembly of more than one protein in one reaction.
Next, I will explain the general procedure and also specific procedure, especially towards the bacterial expression by using E. coli, and also how the interpretation of the procedure. Before we done the specific procedure on protein expression, we need to know the general procedure first. The first step is to obtain the DNA sequence of the gene encoding the protein of interest. This can be done by isolating the gene from a natural source or by synthesizing it chemically. The gene is then inserted into a plasmid or other expression vector, which contains regulatory elements to drive gene expression. Then the clone can proceed into a suitable protein expression system. After screening the positive clone present, the best construct or expression host must be selected depending on type of cells used. For mammalian cells, it needs to expand the transient or stable transfectants, and the protein can be obtained after the cell-based studies carry on. While for bacteria, yeast, and insect cells, it needs to scale up for high. Next, I will explain about the specific procedure, especially towards the bacterial expression system by using E. coli. Because of their fast doubling period, bacteria serve as quick and simple platforms for expressing recombinant proteins. E. coli is the most extensively utilized host system because its genetics, genomic sequencing, and physiology are well understood. Genetic manipulation is simple and it grows to great densities, making it appropriate for large-scale fermentations. The following processes are involved in protein expression using E. coli. First, apply the competent E. coli cells to take up DNA sequences of interest. The DNA sequence is then integrated into bacterial genome or circularized to live as a plasmid. The altered E. coli is then selected by using a selection marker. Expansion of selected E. coli to a larger scale is suitable culture media such as traditional lysogeny broth. Lastly, the protein isolation and purification able get from intracellular or secreted sources. After then, the protein expression, by the presence of elements in the protein expression and consideration of aspects that can determine the protein expression system selection, the procedure should be successful. Therefore, the target protein has the ability to express themselves successfully.
Next is the advantages and disadvantages of the protein expression system, which are specific to bacterial expression system. First of all, the advantages of bacterial expression system is high expression level. Bacterial systems, such as Escherichia coli or known as E. coli, have the capability to produce high levels of recombinant proteins. They have efficient transcription and translation machinery that allows for rapid and abundant protein synthesis. This feature is particularly advantageous when large quantities of protein are required. The net advantages is fast growth rate. Bacterial systems offer quick production timelines for recombinant proteins. Bacteria have short generation times and can rapidly multiply, allowing for fast protein expression and production. These advantages is beneficial when a rapid turnaround is needed for experimental or industrial application. After that is protein stability. Bacterial expression systems can support the production of stable proteins. Bacteria have reducing cytoplasmic environment that helps prevent the formation of disulfide bonds, which can lead to protein misfolding and aggregation. This feature is advantageous for the expression of proteins that do not require complex post-translational modifications or proper protein folding. The last advantages is low cost. Bacterial expression systems are cost-effective compared to other expression systems. Bacteria are relatively inexpensive to culture, and their growth and maintenance in the laboratory are straightforward. The simplicity of bacterial systems contributes to the cost-effectiveness of protein production at large scales. The first disadvantages of bacterial expression system is lack of eukaryotic post-translational modifications. Bacteria such as E. coli do not have the machinery to perform certain post-translational modifications commonly found in eukaryotic proteins. These post-translational modifications include glycosylation, phosphorylation, acetylation, and complex disulfide bond formation. If the recombinant protein requires specific post-translational modifications for proper folding, stability, or biological activity, bacterial expression system may not be suitable. After that is protein solubility issues. Some recombinant proteins tend to aggregate and form insoluble inclusion bodies in bacterial systems. Inclusion bodies are dense protein aggregates that require additional steps for solubilization and refolding to obtain functional proteins. This process can be time-consuming and may result in low yields or loss of protein activity. The next one is potential codon bias. Bacterial expression systems may have codon bias that can affect protein expression efficiency. If the codon usage of the recombinant gene differs significantly from that of the host organism, it may lead to reduced protein expression levels or even translational errors. Last but not least, the last disadvantages of bacterial expression system is incorrect protein folding. Bacterial cytoplasmic environment lacks chevrons and cellular machinery necessary for the correct folding of complex protein. As a result, certain eukaryotic proteins may not fold properly in bacterial systems, leading to misfolded or inactive proteins. This problem can be mitigated by the use of fusion tags, co-expression of chaperones, or the use of alternative bacterial strain optimizers for protein folding.
For the application, protein expression can be used in research and therapeutics. It can be applied for the production of antibodies, enzymes, vaccines, cytokines, and pharmaceutical proteins. The expression of recombinant protein by using bacterial expression system, which is Escherichia coli, has been widely used for many applications including medicines, biotechnology, protein biochemistry, and molecular biology research. Comparatively to expression in living cells, there are many applications that are made easier by cell-free protein expression, since proteins are produced in an open system, such as expression screening, protein labeling, nuclear magnetic resonance, crystallization or biophysical, and further applications such as mutagenesis studies, production of toxic proteins, or enzyme screening. To conclude our presentation, protein expression can be described as a process in which the living organism synthesizes, modifies, and regulates proteins. This process is dependent on the functionality in the cell of the organisms. Protein expression in Escherichia coli is one of the most powerful techniques in research settings, but each of the steps can be challenging to perform. In addition to bacterial expression systems, there are other examples of protein expression systems, which are yeast expression systems, mammalian cells expression system, insect cell expression systems, and cell-free expression systems. All the types of protein expression systems have their own advantages and disadvantages. Bacterial expression systems can produce rapid growth rate, low cost, protein stability, and high expression level. The protein expression can be used in many applications to produce another target protein. This is our reference. Thank you for watching our presentation.